Okay, so good morning all. So today we are going to have a session on using literature to develop the research project. So how we can actually utilize the literature. So I will be explaining using literature and then the process actually that we have developed at Solar Biotech. Uh, so that way you can convert that into a research project where the biomedical knowledge which you gain from the literature can be utilized for the development of the literature project. Let me just share slides. Okay, so as I mentioned that the purpose of this session is to show you the flow that how literature can be utilized into a research project and a discovery uh, can be done. Uh, so I'm Dr. Praveen Bades, Alive Biotech Private Limited's founder and CEO. The purpose of this session is to actually introduce to you the project-based programs that Swalife Biotech, in association with the Swalife Society for Translational Science and Innovations, have created. So before moving towards the topic, I will be talking about three major things. One is about Solar Biotech. Second is about the career opportunities which are available now due to AI. And third is about the project-based program. And then we will be moving towards the topic. So just to give you a brief explanation that what Swalife is doing, we are actually are AI-driven translational science from predictive to preventive healthcare, where we are enabling the AI in the process of this translational research, so that way academic research can be used utilized in industry, and a real world solution which can be utilized in prevention and precision healthcare can be developed. So vision is to actually have a reaction to prediction, so that way healthcare can be moved from reactive to the predictive intervention. Early and safer decision can be made, and accessible innovation can be available. So our mission is to actually have AI-driven platform which can help to connect discovery with the clinic, and while doing that it is already scientifically backed formulations, and it will have the solution there. So while doing this, we actually have this project-based program where students or the faculty or the researcher can join us and work on those projects. So we're not just generating the data; we actually making it for the decision, and how we will be showing that. The core values which we are creating is that we are making sure that the evidence-based thinking is there, science first, where we are making sure the AI is supporting our human intelligence, so that way we can actually translate the research for the world use. And clarity is being utilized to have those innovation and affordable, practical, and scalable for global market. So why it is mattering you? Because with this project-based program, you will be like you will be thinking like a researcher. So that is what one of the major um, One of the major skill you will be developing. Simultaneously, you will have the access of tools of Solid. So that way you can understand that how the AI is actually helping us to bridge the gap between the discovery and practical application. So you are learning how industry works. So these are some of the projects which are there at the moment, which we will be starting from March, majorly with the AI and with the large language models. The targets, the leads are already been utilized, and you will be working on them. So there are majorly 5 projects, and we actually have limited seed for each project. So when we look at the career opportunities which you will have by learning the skills uh, which are in collaboration with AI. The major first one is the drug discovery and R&D roles, like research associate is there, R&D executive is there, discovery scientist is there, and also translational researcher assistant can be some of the roles which are there with them uh, gaining this skill. Then AI enabled life sciences roles are there where AI in drug discovery for analyst purpose, then AI research assistant and computer biology training. So when we are talking about AI, it is not going to actually completely replace human. Yes, some part of the human work will be replaced with the AI, but the human who can actually you know, can handle the AI will be more um, you know powerful and can be one of the major um, skill that person having is that he is utilizing AI as assistant and working with the help of that AI. So it is very important that in life sciences AI is going to be there, and those roles will be filled by those people. Next is clinical research coordinator, clinical data interpreter, associate, translational research executive, and clinical strategy support. So these are again bridging the science to patient care, and here predictive analytical predictive medicine is going to be playing important role, and AI is actually helping in this role. Uh, the old thing about pharmacovigilance drug safety is now changing. The roles like pharmacovigilance associate, then signal review trainee, all this is now utilizing AI to understand that what signals are available for certain drugs or certain diseases, and that skills need to be learned. Then drug safety analyst, safety documentation executives are important opportunities. Then with the regulatory affairs that we know now, the documentation for regulatory is gone digital now. So ECTD is there, then clinical summary, regulatory intelligence, compliant documentation. All these yeah, roles, all these skills need to be there so that way regulatory affairs trainee can work on them. And scientific writing, the person can do the dossiers preparation and summaries of the clinical data, so that can be drafted for publication. Then regulatory document associate can actually create ACTD. So this is then separating in which opportunities can be utilized using the skills which you can get from the project-based programs. Research writing, publication support, literature searching, reading, and evaluating is the major role of the researcher. So scientific writing writer can actually create clinical document. Research analyst can actually synthesize and interpret research finding. Publication support will be needed for manuscript preparation, and review article writer like grant writing and other things. In addition to these opportunities, you can also work as an entrepreneur. You can start your own startup innovation for R&D innovation, product development, health tech, where scientific validation skills can be utilized. The next is that higher studies, where you can actually look into applying research internship at international opportunities which will help you to get fellowships for masters or even for the PhD. So the major key advantages which we think as a company we want the student or the faculty or the person who want to gain skills in AI is by actually working on real project experience which are utilizing AI and doing the translation of the science into the decision making. So uh, this is an industry aligned certification where you are going to be recognized uh, from the industry. So majorly, I have discussed these skills, which you, these opportunities, career opportunities, this based on the skills which you are going to gain. So what is the basically the project-based program which we have designed? You will be working on a project which I have mentioned earlier. You apply the concept using AI tool, and there will be a scientific workflow and a real research that how actually the research is going to be done in the industry. You will be part of it. So one complete end-to-end research project will be given to you, where you start from literature mining and you move towards predictive medicine, which gives you clear understanding that how the idea from science to decision making can be done. In this process, you are going to have literature. Different tools are available of Solid Biotech, where literature mining till predictive medicines. You will have the experience. So in this process, you are gaining the research thinking skill, where hypothesis building is one of the major important part in this, and also the research methodology. Then, with the help of the network pharmacology, you will understand drug target interaction. With the AI assistant tools, you will understand the target and the mechanism of the target using the tools, and you will be then moving into the predictive analyticals, where you will be interpreting and taking the clinical decision using the logic. So you will have the access to the research platform developed by Swalab Biotech, from literature mining till predictive medicine interpretation, where clinical decision supporting framework will be designed. So we'll have a structural mentorship where weekly goals will be there, feedbacks will be given, deep understanding about the topic help will be given, and the expert guidelines will be there. Guided sessions will be taken weekly. Your goals and output will be clear. Feedbacks will be given, so that way you can focus on why and how and not just what. So the deliverable in the project are complete project report, uh, AI research insight will be there, workflow diagram, and final presentation. In addition to this, we want two things out of it. One is the review relevant to topic and the work which you have done. There will be original article from that data. So you will be getting the official certificate from Swalab Biotech as you complete the internship in AI-driven network pharmacology and predictive medicine. Who can join this program? Majorly the student, the scholars who are doing postgraduate and PhD. Also the early career researcher who want to understand the AI, and the professional who want to explore AI in healthcare and drug discovery. Uh, in this case, you don't need actually the coding, and now with the vibe coding, there is no need actually to learn the coding. With the scientific prompting, lot of things you can develop in this process. If you develop any idea, we can take it further. So the program duration which we have kept is 4 week, but it is not just 4 week. You can continue on different projects every month. So there is no limitation if you want to go much deeper into predictive medicine, into preventive medicine, or into precision medicine. But for this project, it is 4 week. We'll have sessions every week, 3 times, so that way you can understand the science behind it as well. And one session out of three will be question and answers, where you will get the feedbacks if you have any question which will be resolved. And it is completely online with flexible schedule. So the recordings will be there. You will also get the recording. At the moment, the program fee we have kept is 999, so that way it can be affordable to everyone and everyone can have the access. So what the cost cover, training mentorship, the AI platform access, the project resources, review and feedback, and with official certificate. So simply real research project where AI and life science integration will be there. Industry relevant skill which help you to develop the strong portfolio certificate from Solar Biotech at the affordable practical level. So if you have any questions, you can add in the comment section. But we will continue now to the next part, that is the main project, main topic of our session today. That is the literature to the discovery. So when we look at the reality of this modern biomedical research, we can see thousands of paper are there, but still we don't have the clarity. There is a limitation of clarity with gaps between this research and the real world application, which is actually the major challenge in the converting the research into the translation medicine. And that is happening due to this increase in the volume of this publication. Why? Because the journals and the funder majorly rewards the novelty over defining findings, or even small underpowered studies. So majorly the results are flooded with lot of positive results, and it is difficult to convert them into the actual product. So the reproductivity crises are there. It is difficult to replicate the same papers. So major of the papers are unable to reproduce. Then the second thing is we look at the academic, we look at the company. There is not in the company, but at least in the academic where the major publishing happen. There is a pressure to publish. So weak methodology has been utilized to make sure the articles will be coming faster, and incomplete reporting is there. When we look at the methods, lot of things are missing, and it is difficult to utilize those methods and replicate the results. So reproductivity crisis of the research is one of the another major thing. Then the barrier to implementation. So individual level, you cannot actually take it further. With the organization level, there is a lack of resources, equipment is the issue, weak leadership support is there. And third is the system level where guideline lag behind the evidence. So there is no proper guideline regarding the research, regulation, and all this misalignment is there. So there is a barrier to the implementation. So if I give an example, like when it comes to the formulation, nano formulation, a lot of people think that this is a one of the area. Definitely it is one of the great area to do research, but when it comes to bring it into the market, there is a big barrier, and that barrier need to be understand at the beginning when you are actually moving into the area. The bench to bedside is a big gap. When we look, roughly one third or more of the healthcare delivery is not actually aligned with the best evidences, and that's why there is a gap, which is difficult to convert into the into the bedside or into the drug due to that gap. So when we look at the translational research stages from the discovery to early trial, it need to move in proper way, so that way early trial can be done, which will be then moved into definitive trials where large scale efficacy studies can be done, and guidelines and implementation, that is translation of those evidences into the practice, can be continued. Then there will be continuous feedback happening, that is monitoring the outcome and the care, so that way it can be refined further. So bridging the gap is the important point there, which need to have solution, and that solution is improve research quality, make research usable, focus on implementation, and build capacity. So better designing will help, clear summaries in the article, having proper methodology in the article, structural abstract will help. Also having tools where non-expert can understand the methodology, and also the limitation can be a solution. Co-designing with clinician and the patient is actually again a major focus area. So that way it can be aligned with a proper context, and training professionals so that way they can create knowledge which can be connecting literature to practice. So there is a uh, bridging that gap is important. So when we look at the literature, so whatever we discuss at the moment shows that literature alone is not enough. So understanding now, when we look at the evidences with the clinical studies, we will see that two types of reviews are there, like narrative review and structured review. So narrative review is actually giving you history, theory, and opinion, but lacks systematic rigorousness, where systematic review is actually having a predefined process like defined questions, search databases, screening studies. So then it actually helps to uh, understand the reproductive productivity of the articles which are available. So use narrative for orientation, but rely on structural evidences for decisions and protocols. So that's very important when we are actually working with the literature. Decision centric understanding is important because patient subtype, pathophysiology, and population difference need to be understood. So, clinical labeling often bundle with patient, where deep mapping of diseases mechanism help us to understand the pathways. But when we are doing this, the population difference, so the paper which is being published in certain areas can be for the certain population only. So mechanism can vary based on where actually it has been done. Molecular level clarity is also important to understand the target in that specific cell, so that way it can actually help us to translate into the efficacy into precise intervention strategy. So when we look at the evidence structure framework, questions and dimensions help us to develop the strength assessment, which help us to understand the gap identity. So it takes from chaos to clarity. So evidence structure is very important to come towards the priority areas for the research, so that way the gap is being identified. So evidence map and gap analysis is going to help us to take towards the proper research question. So visual mapping show where the evidence is strong, weak, and missing. Need to be done, which help us to guide both clinical decision and also future research investment. And it also give us the transparent logic, that is the trace recommendation back to specific evidence score and quality assurance. So that's where these things is leading with the help of the literature. When you actually focus on narratives and review and systematic review. So if you have any question, you can add to the comment box. I'm just checking comment box. No question. Okay. So when we start, the starting point will always a disease biology. So that way we will understand uh, that how actually the disease has been created. We are not going to start with the tools. We need to understand biology of the disease first. So why disease first? Because understanding the pathophysiology, that which cell, which pathway, and which process-driven progression is actually the reason for disease is very important. So the foundation start with the pathways. Networks are connecting the genetic, environmental reason, and also the clinical phenotypes, which is revealing the druggable points. Then comes the targets, where specific molecules using modulation is predicted to alter the disease course, and the biomarkers, which help in the measurable indicators for diagnosing, staging, and the patient selection. So aligning this drive actually help to develop the hypothesis. So when pathways, target, and biomarker align, you can design study testing for that specific hypothesis, and this aligned transfers into hypothesis-driven science. So disease-centric mining is important. You mine the existing knowledge, so that way you get exact genes, pathway, variant, and phenotype and that is from the literature and the database. So that way you can build disease-specific network showing the mechanistic relationship, and also prioritize the target. Using those network, you can rank the target and the biomarkers instead of just scanning the targets. Now, this also help us to enabling the repurposing. So, when we talk about repurposing, once disease mechanism is understand, you can match them with the known drug, similar diseases or shared pathways. So that where disease-centric approach to drug repurposing can also be done. Now, drug repurposing is one thing where the drug is already in the market. Second, you can also go for synthetic molecules. But in third, where we are working on is the plant and the secondary metabolites from those plant, because it is a chemical ecosystem, and also secondary metabolite or plant extract is not a single drug. So understanding this complexity help understanding the metabolite mining. So plant is not equal to single drug. It is actually chemical complexity. Numbers of secondary metabolites are there in that plant, which help in the disease. So understanding that mixture interacting with the biology of the human is one of the very interesting subject, and that's where the solar biotech is doing at the moment. So just using the traditional mixture is not what we are doing. We are actually making sure that the single molecules from plant in combination in extract format are focusing on the targets, and then we are identifying that target, that how they are actually helping in the disease. So secondary metabolites matters. Why? Because defense against microbes or pollen, they are already working with the plant. Then when you translate them into human, they are actually useful in antimicrobial, anti-inflammatory activities, which has already been well documented. And major drugs which are there at the moment in the market has been from the sources of plant secondary metabolites. So when we look at the crude extract and isolated molecule, as I mentioned earlier, that this is what we are doing, and these are the two major approaches at the moment where a lot of companies are working. But when we look at crude extract, it actually have got higher complexity, and it gives you low clarity. But when you do that with the isolated molecule, the complexity is reduced, but the clarity is increased. Now, they got their own pros and cons. We have seen that at Solar Biotech as well, that synergetic activity of the extract sometimes is high compared to the isolated molecule. It can be due to the combination of different molecules. The molecule in the extract who are actually having very less uh, concentration can also plays important role when they are there. Same thing with the isolated molecule, due to those molecules which has been removed from the extract. Due to that, the activity can be effective. But then there is a process called deconvolution, where you can actually use crude extract, but you can fractionate them, and those fraction can be tested and identified. Now, instead of taking one molecule and then for the plant, we can actually have the fraction. So the small molecules in the fraction can be still you know, showing activity due to the synergetic effect of having those compounds. And we have done this. We actually utilize fractions from major plant extract, which has already have activity in a have product, but we fractionated and then we actually using them in the product. Now, while doing that, metabolic identification is one of the major step in this. Now, when we look at this and from for this types of project, it is more about pharmacognosy, but simultaneously the chemistry comes when you are identifying the metabolite, because this metabolites plays important role when they are in the combination, because the synergetic effect is there. In addition to that, those metabolites are linked to the targets which actually are mutated, or there is a issue in the human. in those targets. So those targets have been focused by these leads, and these leads, when they are identified, can actually give us information about which leads have targeted which protein, and the drug likeness property can be identified. So it is very important to identify those leads. Now, connecting plant to pathway is one of the, as I mentioned earlier, is important part where you can make them disease-centric mining plus metabolite identification connection. And once that is done, you can then move that plant X is good for disease Y. Why? Because the targets has been hit by the molecules from those plants. And there where actually we are into the network pharmacology, because now we can actually create a networks of drug that is the lead from the plant with the biology that is working, and then the interconnection network can show that which leads is hitting which target. So one drug, one target has been now not the only one way to develop the treatment and the formulation, but having numbers of targets hit by numbers of molecule is an important player, because synergistic effect of those leads is a major um, major and important part of the formulation of the drugs. So why complex disease need network? Because as I mentioned, that numbers of targets are playing important role in the disease. So it is very important that those need to be understand, and alternative, the single drug is not going to hit all the targets. So multi-target therapy need to be created, so that way numbers of molecule from the formulation can hit numbers of targets. So the network need to be understand before actually moving into multi-target therapy. So network level intervention is something where you analyze the disease network for herbs, bottleneck, and vulnerable motifs. So that way you can design a combination therapy, and a pathway level targeting can be done. So building a network from literature is what the first step, where the drug compound targets pathway disease. All these can be utilized to have nodes and the edges. So in the edges you can understand the relationship extracted from the database and also from the publication. Then combining curated database, so that way you can integrate the data source and analyze with graph therapy, where finding key target and multi-target intervention can be set used by building from the literature. There are different type of networks, and it is important to understand them. So you can actually have drug component target disease network. You can have disease gene target drug network. You can have target pathway network. So this plays important role, like plant compound connection can be developed with the first network, drug repurposing done with the second network, or mechanism analysis can be done with the third network. So from plant to network, we are moving using the literature, using secondary metabolite from the plant and the network from plant. So secondary metabolite have been identified, which help in predicting the targets and validating the target. So that helps to understand the disease pathways. And once that is done, you move into the network analysis. So this is what we are actually doing into our program-based, project-based program. In this tools which we have developed actually help us to understand all this. And also network pharmacology using different databases, which is open source, will be also used developing the AI based formulation. So if you have any question till date, as said earlier, you can put into the uh, comment section. Okay, so continue to the next part, is where now AI will enter the discovery. So with the help of the tools, with the help of the network pharmacology, we have got the information and also the data which we need to put into now structural data, but simultaneously in the patterns, also which can help in proposing ideas, and that ideas can help us to take the next decision. So now AI is helping to understand the pattern recognition. So there are models which can actually scan huge amount of data sets, and that can help in putting uh, the data into a relationship or the associationship between disease target molecules. So when we develop the network, that network is giving us information about different target lead and the disease. But with the AI, it helps us to understand that this is the pattern for certain types of molecule category, and then we can actually utilize it further to understand that a successful therapy can be developed using those category of the candidate or the leads. It also help in generating the hypothesis where, based on the literature, the data which you have got can be converted into the understandable or understanding the gap which can help in the hypothesis. In addition to that, there is a generative models which are there in these large language models, which help us understanding the structure of the molecules, the combination, the protein-protein interaction, target combination, and also when we are moving into clinical, it helps to understand that what type of patient groups can actually be utilized. So all these types of understanding can be given by the AI, the large language models, uh, while generating the hypothesis. Now, one of the major thing is that can be utilized as a decision support. So some of the tools we are developing for clinical purpose are actually playing this role where they are helping in the decision support. So target prioritizing. So when we look from the discovery point of view, from the uh, research point of view, the target which we have identified, we can actually score them, rank them using AI. Then the molecules which we have identified can be actually utilized, or they can be synthesized. The modification can be done. In addition to that, we can go for combination, multi-target, multi-compound combination. And as I mentioned in the clinical trial for precision medicine, they can help in the decision support. Now AI is assistant. They're actually helping us to generate the uh, you know decision making where what to do next. They are not actually going to do next thing, but they are going to help us by working as a pattern recognizer, hypothesis generator, and decision support maker. And this assistant is going to be powerful when we are actually moving from the literature to the research question. So when we are doing this transition from literature, we actually have put one of the AI discovery studio that we have created, which help us to build the network. It help us to run the AI models, so that way we can actually have candidate the predictive biology. Once that information is there, we can actually have interactive inspection done, so that way that AI result can be validated. And once that is done, we can curate the literature. That way we can convert that paper into knowledgeable database. So disease biology is the first thing, and then based on the disease biology, we moved into AI to mine, connect, and prioritize the molecules with those targets which we have identified using the disease biology. So I am now also talking about the stages which we will be having in the project-based program at different stages. So stage one, which we will be having, is that we are curating and structuring the literature. So from the text we go into, so text is unstructural data. We move into the structural data, and that while doing is done using the tool. So we build the structural knowledge database here, so that way it is used further into the generation of the hypothesis. And with the help of that, we build the disease and compound network. So whatever we have put into the structural database is then moved into a network formation, which construct by using disease gene and pathways. And those is one network, and the second is the compound network, where compound target network is done. So this happen in the first week of the process, actually first and some of the second week part. Then we run the AI models. So we use the network, the compound, the literature uh, into our AI models where graphical network or machine learning tools are there. Also the LLMs are there. And then those outputs are that rank targets are there, predictive interactions are there. In our case, it is the rank target and predictive interactions. But if you're also working on drug repurposing, you can actually have the repurposing candidate information. So AI is utilized so that way prediction can be done with the disease biology, and that happens when we started. Now in stage 4, we interact with Discovery Studio. So there are a couple of models in the Discovery Studios which help us to explore pathways, compound, and hypothesis testing. So this information we get. Now this knowledge need to be converted into discovery. So structuring those knowledge actually need to be then convert scientifically into discovery, because sometimes AI actually make hallucination which are actually some of the time they are not there but it has been generated. So then this structural literature with the AI logic, now we need to put them into proper foundation. So structured biomedical graphs which have been created of disease gene pathways need to be put into a network where nodes edge can actually give us the features of those network. So graph and machine learning models can utilize to understand this pattern. So when we look at those patterns, we are understanding the disease pathway, building those initial gene disease pathways, then creating the features, and then we are enabling AI so that way it can give us the patterns, so that it can give us the ranking, proposition, and suggestion. So data quality actually determines that how AI is going to work. So we need to make sure that whatever data we are generating and the network we are generating, it is of high quality and it is from the genuine source. So if you put the unstructured data, it is going to give you the poor or even the unstable, unpredictable, uh, interpretable predictions which are not going to be relevant to what we are trying to do. So it is a critical and very important thing that you actually have a quality foundation which has been curated by data which has come from the proper source. So that way AI can actually give us the proposal which is then explainable and also in a critical way, which we can actually validate it because it actually have a clear data curation done. So it is important that we actually focus on week 1, and this week 1 then actually make our week 2, because you actually now got disease gene pathway tables there, metabolite target networks there, biomarker catalog is there, and also literature evidence mapping is done. So that helps us to move into week. So the complete pipeline from literature to AI driven through systematic structuring will happen. So the journey start week 1 understanding, week 2 structuring, week 3 mapping, and week 4 identifying. So each stage actually build on the previous, so that way creating a foundation for trustworthy AI discovery will happen. So the core principle here is that the disease first, so that way you understand the disease, and then the structural evidences can be actually taken from the literature where decision can be done by the AI, and the networks and nodes will be created from the literature which will have a quality over quantity. So we don't want the quantity. So if we take an example, let's say a common plant like tulsi can have millions of paper, but if you look at the quantity and quality, it is going to create a quality matters, not the quantity. So for mission, week one's mission will be build the foundation that is about the disease, and then we move into week 2 where AI experiment the tools which we got. We start working with them, so we validate the the data by deploying AI intelligence. So we rank the target, predict the interaction. Then we also do the query to the AI Discovery Studio so that we can test the hypothesis and we validate the experiment to confirm that the AI suggestion that we have got in week one can be utilized further. So we are not only relying on the data which is there from the AI, we put our human intelligence to make sure that what we have got can be taken further. So if you look at the success rate, it is about 100% because whatever um, prediction done, we can actually get top candidate that is about 3 to 5 candidate, and we can develop one proper hypothesis. Why? Because we are actually looking at the complete quality data which is in text format, and then it is converted into the structural format. So the common pitfalls which we need to avoid while doing this is that we are not going to look at tools first. It is the disease understanding first. Then we are using the tools. Skipping structure, you need to go as we have actually done previously. So that way you are going to start with the raw literature and then utilizing AI. Single target we are going to avoid, and then the black box AI, that is what I said, that we are not just going to utilize AI understanding, but we will be also putting human intelligence so that way you can validate what we have got with from the AI. So your project roadmap is disease track, then secondary metabolite, and then the AI. And they all progress to make sure that validate hypothesis is being developed. So the questions to guide you to work is that disease selection, plant choice, network design, and AI queries. Now all this is already been done, and we actually already have those project listed. I showed you the list previously. So this actually help us to bridge the gap which we mentioned earlier that it has been created due to the literature. So this approach actually help us to create the hypothesis where the biomedical research and real world impact can be combined. A structural AI powered pattern recognition can be done. So to begin with, week one, disease pathway, metabolite, and building network is what we will be doing. Then move into week two, week three, and week four. So this is about the literature towards biomedical research using the uh, data from the literature in the text format, which is in unstructured, is converted into structural format, and that is helping us to fill the gap which has been created due to the literature, and a research project can be designed using only the literature. So that's about literature to discovery and how we can actually convert that into a research project. So if you have any questions, I'm happy to answer. You can put your questions in the comment box. Okay, so if you don't have any questions, I will see you later. If you got any question, you can put in the WhatsApp group or you can share with my team. Thank you.